Good evening, my dear friends. I welcome you all to the Hindu Science and Technology analysis on 28th August 2022. Today, I am going to discuss which is not part of the science and technology column of the Hindu, but which is of relevance today as the current news. That is, Supertext, the towers, the twin towers, as part of the Emerald Court project, with Apex of building with 32 floors and Sian building with 29 floors have been grounded by controlled implosion. The buildings, the twin towers, Sian and Apex, have been grounded because it violated the Uttar Pradesh apartment regulations 2010. The Noida Authority, that is, the Noida Authority had allotted the Emerald Court complex of constructing 14 towers of 9 floors each, a garden area, and a shopping complex in 2005. However, in 2009, this was altered to make way for the twin towers Sian and Apex in the garden area with the area plan for the garden area, which attracted the wrath of the association, residents welfare association, who complained to the High Court, and the High Court ordered the demolition of the flats in 2014. The plea was moved in the High Court in 2012, and within 2 years, in 2014, it was ordered to be grounded. The Noida Authority and Supertech Company moved the Supreme Court, and on August 31, 2021, the Supreme Court ordered the demolition of the flats and upheld the High Court order, the Allahabad High Court order ordering demolition of the flats. So we have seen that today. Actually, the Supreme Court had ordered the demolition of the flats within 3 months from the date of the order. That is, August 31, 2021 was the order. So within 3 months it had to be demolished, but it took nearly 1 year. To demolish the flats, it took nearly 1 year. The project of demolition, which is carried out by the Edifice Company along with South African jet, Mumbai based Edifice Company along with the South Africa based jet, which had carried on the demolition of 4 tall structures in the Marat flats, that is the river looking Marat flats in Kochi, just a few years ago. This technique, this controlled implosion, the technique is known as controlled implosion and was first tried in 1773 to ground the twin towers, the Trinity Towers Cathedral in Europe, in Ireland, in Europe. The Trinity Towers Cathedral was grounded in 1773 in Ireland, in Europe. This was the first time the controlled implosion was carried out. So, how this was carried out? The blast detonators were used, electric and non-electric, to initiate the blast, and emulsion, which is used in the underground mines for the hard rock grounding, blasting. Emulsion was used in a very low dose in this structure. In a very low dose. 6,500 kilograms of emulsion was used. Then the shock pipes, the shock tubes were used for enacting or the initiating of the blast. So, 3 items were used: the emulsion, which is the explosive, the shock tubes for initiating the blast, and the detonators, electric and non-electric detonators were used for blast of the twin towers of the Supertech building. The Xi'an had 11 primary blast floors. That is, it took 7 months of planning and carrying on of the preparation for the site of the blast. 1 month of planning was used and 6 months of strategically placing the explosives in the columns of the buildings was used. In the 11 floors, the base primary floors of Xi'an, the explosives were placed in almost all the columns. In the apex, there were 10 primary floors where the explosives were fully placed on the columns and 7 secondary floors. The 65% of the explosion was triggered by the primary floors where the blast explosives were placed. Therefore, there was a doubt whether this would create the shock waves around the area, around the region where these buildings were located, because there are nearby buildings also. The For making it a very smooth process, the 13 seconds process of the blast, out of the 13 seconds process of the blast, the final site, that is the final item, the final cornerstone of the edifice was grounded around 7 to 9 seconds after the first portion of the building was grounded. So, this has obviated the necessity of all concerns that it would create a huge shock throughout the area. Around 500 meters were declared as no entry zone for animals and human beings. All animals and human beings were evacuated from the site, and only the police personnel, the NDRF teams, and fire force people were present. For flouting the building safety regulations and fire safety regulations, this building was grounded, and it has produced around 80,000 kgs of debris, 55,000 kgs of which would be used on the site to fill the site, and rest will be used for processing in other sites. This is the largest demolition of its type in all over the world, and it has happened today, has which we have witnessed and seen. This shows that flouting the regulations of the urban authorities, the reckless building of the reckless constructing of the buildings, defying all ecological senses and regulations, is a cause of concern, and this will not be entertained. Friends, the next article I am going to deal with is the study of nuclear matrix with the help of the fruit flies. The fruit fly genomics is used for studying the nuclear matrix found in a cell. Nuclear matrix is just like the structure of a building. The building becomes inhabitable only when we move the furnitures inside the building, the electrical fittings inside the building, and the sanitary wires inside the building. In the similar way, in the 220 types of cells in the human body, there are 220 types of cells in the human body. One cell is for the neuron, one type of cell is for the neuron, another type of cell is for the intestine, another type of cell is for the liver, another type of cell is for the skin, another type of cell is for the tissues. So different types of cells, that is 220 types of cells are there in the human body, and each cell is containing a nucleus, and the nucleus contains chromosome, and the chromosome contains the genetic materials. It is called a genome. The genetic materials are packed in the nuclear matrix, just like the furnitures are kept in the building. The genetic materials are kept inside the nuclear matrix. And when a person is coming to our house, he rings the calling bell. Only when we open the door, he enters the house. Similarly, the transcription the transcription material, which is responsible for carrying the genetic material from the nucleus to the cell development proteins, that is ribosomes, is trying to access the genomes. The genome, only if it allows, that is it is allowed with the help of the nuclear matrix. The nuclear matrix is the door. Only if the nuclear matrix allows this door to be opened, then the transcription material gets access to the genome. Why this scaffolding is necessary? This scaffolding is necessary because in the intestines there is a protein. The protein which is being just prevented from being corrupting or destroying the intestine, the protein is called protease. This protease is prevented from eating away the intestine by the nuclear matrix scaffolding. That is, the nuclear matrix door or the structure is preventing the genome to be destroyed by the protease. The genome of the lining of the intestine to be destroyed by the protease. Protease protein, if there was no nuclear matrix, it would come out and destroy the lining of the intestine. Therefore, in order to prevent this, the nuclear matrix is preventing the interaction between the protease and the lining of the intestine. Similarly, in the brain also, this protease is present, and this protease, if it interacts with the brain cells and the nerves, it will destroy the brain within few seconds. Which is preventing this destruction, my dear friends? Nuclear matrix. This study of nuclear matrix is very important. Hence, the fruit flies are used. The fruit flies are scientifically known as Drosophila. These fruit flies embryos are taken and the DNA is treated with an enzyme. The biochemical process is carried out in the embryo, the nucleus of the embryo of the Drosophilus fruit fly, and the DNA is treated with an enzyme. And when the DNA is deactivated thus, and another salt like solution is applied to take out the nuclear matrix. This is the biochemical process applied to take out the nuclear matrix, to study this nuclear matrix. Hope you understand, my dear friends. So this nuclear matrix is studied with the help of the cell of the, that is nuclei of the cell of the Drosophilus, that is the fruit fly. Friends, next article I am going to discuss how the mosquitoes are detecting the human beings. August 20th was the mosquito day. There are 3,000 mosquitoes in the world, and among them the most dangerous ones are the Aedes aegypti, which is causing dengue, the Anopheles, which is causing malaria, and Culex, which is causing West Nile fever. So, how does these mosquitoes detect a human being who is 50 meters away? The mosquitoes are detecting human beings who are 50 meters away because of the olfactory neurons present in them. These neurons inform them about the human presence of human beings by the chemical found in the sweat, that is 1 octane and 3 all. 1 octane and 3 all chemical found in the sweat informs the mosquitoes, the olfactory neurons of the mosquitoes, that the presence of human beings is there. This will work 50 meters away. So, what we do is that in order to inhibit this olfactory neurons, that is, prevent this olfactory neurons from detecting human beings, we use repellents, mosquito repellents. What does this mosquito repellents do? The mosquito repellent is having the chemical DEET or diethyl toluamide. DEET, diethyl toluamide, is a chemical found in the mosquito repellent. This will obfuscate the olfactory neurons, that is, odor receptors. Olfactory neurons are passing the information to the mosquitoes through the odor receptors in the mosquitoes, which is found in the tentacles. The antennas of the mosquitoes carry these odor receptors, which will catch the smell of the human beings 3 or 3 octane 1 all chemical in the human beings and inform the olfactory neurons and then detect the human beings. This diethyl toluamide chemical present in the mosquito repellent will inhibit, that is, obfuscate. The term is called obfuscate the mosquito's antennas. When this diethyl toluamide is acting on the antenna of the mosquito, the odor receptor becomes dead, not dead, temporarily inactivated. So it cannot detect human beings. However, this is common for all animals. However, the mosquitoes are still biting human beings. What is the reason? Recently, scientists have discovered that apart from one chemical which they are able to detect of the human sweat, there is another chemical. The name is amine, AMINE, which is also present in the sweat of the human beings. Amine present in the sweat of the human beings can also be detected by the olfactory nerves of the mosquitoes. How this is possible is not known. When a secretion is made by the human body or the animal's body, it will prevent another smell to be combined or mixed with the chemical produced by the body. The cell which is producing this will create in such a way that no two odors should be able to be detected by the olfactory nerves or the odor receptors of another animal. How these mosquitoes are doing this is a cause of concern. The mosquitoes are able to detect not only 1 octane 3 all chemical from the human beings but also the amine chemical of the human beings. This is very strange because, as I said, when the chemicals are produced by the body, it will see to it that no two chemicals are detected by one olfactory nerve or the odor receptor of another animal. Only one chemical at a time can be detected by the olfactory nerve or neuron or the odor receptor of another animal. This one to one connection is broken by the mosquitoes. How this is happening is a very cause of concern. We have to discover so that we find some medicines which will prevent these both chemicals to be detected by the super mosquitoes who are very much capable in detecting two chemicals at a time from the sweat of the human beings. Then I am going to discuss about the antimicrobial resistance. Once, at the Nobel awarding ceremony, Alexander Fleming, who discovered the antibiotics penicillin, who had discovered the penicillin, had predicted that one day will come when human beings will use anti-penicillin at a very cheap cost and will develop resistance to this penicillin. True, we are taking medicines and medicines are having effect on the body. The medicine contains some chemicals which will create the microbial resistance from the body. However, human beings, what they do, especially in India, take some drugs for some days and discontinue it. And what happens is that super bugs develop which will resist these medicines. So, generic medicines are not working anymore. So, what happens is that the antimicrobial resistance is what we call. Whatever drugs we take, our body has developed resistance to these drugs. Those super bugs are developing. And super bugs are attacking human beings, and this process of resisting the microbial resistance is happening to the drugs. And it is killing around 1.67 million people all over the globe. It is going to become the next pandemic, my dear friends. Just like COVID, it is going to become another pandemic. So, what has to be done? India, the CSIR, the ICMR has to partner with Global Antimicrobial Drug Research Laboratory so that the medicines can be developed, the antibiotics can be developed, taking into consideration the antimicrobial drug resistance. This is the point which is emphasized by this article. And then one thing can be developed, the diagnosis systems can be developed. What happens in India is that for any kind of fever, the medical professionals are issuing just the drugs which resist the virus, which happens that even for bacterial infections, the drugs which resist the virus is being given. So what happens is that the antimicrobial resistance is developing. During the times of COVID, we have seen that we can develop better diagnostic tools and methods. So it is fast developing. So why can't we develop better diagnostic methods so that the drug area, that is drug targeting, can be better affected? Drug targeting means identifying the area where the medicines have to be applied. From the question corner, I am going to discuss the stories of Jezero crater in Mars, which is discovered by the Perseverance, the rover Perseverance. Jezero crater has thrown several insights into the development of life on the Earth. While visiting Jezero crater, which is located nearby a lake, a river, Jezero crater, which is located near a river, found by the rover Perseverance, we had understood or expected that sedimentary rocks would be found. However, to our dismay, we have observed that volcanic materials are found on this Jezero crater. Jezero crater has been there around 4 billion years ago. However, in our Earth, we have found that whatever craters or rocks were found 4 billion years ago have formed a transformation by tectonic forces like earthquakes, tectonic activities like earthquakes, and the combined forces of water, air, and wind, and several ecological or human interventions and animal interventions. This has formed as a sedimentary rock and is found at the bottom of the river. However, to our dismay, the Perseverance has found that Jezera crater has only the material, the volcanic material, that is olivine. The name of the material is olivine. In the Jezero crater, the volcanic material olivine is found, which shows that the tectonic forces, the earthquakes, and all sorts of tectonic forces have not influenced this crater. So it will pave the way for studying the origin of the life forms. It is believed that life forms have originated in are found in Mars, and it is believed that this Jezera crater will pave the way for studying the origination of life forms on the Earth, because this is not disturbed by any kind of tectonic activities like earthquakes, and obviously Mars do not have that much faster winds and the another sorts of water and wind forces which will change it. It has been seen, the Jezera crater has been seen as mostly unchanged. So this is a remarkable discovery by the Perseverance rover,